Well, good afternoon, everyone, and welcome back to the last session of this Researcher Day. Hope you all had a really nice and strong cup of coffee and are feeling full of energy to listen to the last talks. We will continue on the topic of biodiversity, and this time zooming in on the Archipelago Sea, which is, of course, fascinating. Last week, I had a chance to travel through the Archipelago Sea on a ferry. And it every time it strikes me as an absolutely stunning area with massive amounts of habitat as well as biodiversity, both above the surface and under surface. And as our first keynote speaker, we will have the pleasure to listen to Ilari Saksjärvi, who is professor at the Biodiversity Unit at the University of Turku. And he usually works in more tropical areas, but today he will be talking about how to tackle biodiversity loss in the Archipelago Sea. Welcome on board, Ilari.
Thank you. Okay, thank you very much, and thank you for the invitation to join this seminar today. I'm very happy to be here. As said, my own background is a little bit in more tropical conditions than in Finland, but I will try to tell you something about how to tackle biodiversity loss in the Archipelago Sea region. It is very nice to be part of this seminar today. So, as said, I have been working for the last 20 years or so, mainly with the tropical biodiversity issues, and also a little bit with global biodiversity issues. So for the last 20 years, I have been working in Amazonia and also in tropical Andes, and a little bit also in Central American tropical forests. And my own research team has been concentrating on discovering new animal species for science. And we have been quite successful, actually, in finding new species. As you all know, major part of biodiversity is still unknown. And for example, in the biodiversity unit in the University of Turku, we are describing almost 100 new animal species every year. And one example can be seen in this slide. There is Umanella kiekometti, one gigantic new wasp species that we found from the Colombian Andes, from the tropical Colombian Andes. But besides of these tropical biodiversity issues, I'm also principal investigator of two very large multidisciplinary biodiversity consortiums, Biodiforum and Biodiful. And Biodiforum is a large consortium that is trying to promote the systemic transformation across the society that is needed in order to tackle biodiversity loss. And then Biodiful is another large consortium. We have about 20 researchers working in the Biodiful consortium. And in Biodiful, we are trying to study the future leadership. How should we change the leadership in order to save the world, or have a little bit better word for biodiversity in the future? And what is interesting is that both Biodiforum and Biodiful are actually also working in the Archipelago Sea region. So we are just starting our field season also in the Archipelago Sea region. We are doing kind of applied biodiversity research. So we are not collecting, for example, samples, but we are doing a little bit more applied biodiversity research in the Archipelago Sea region. But of course, my background is also in the Biodiversity Unit, which has many links to the Archipelago Sea. Biodiversity Unit is a quite large unit. It's one of the latest departments in the University of Turku. It was established only about 5 years ago, and we have about 70 people working in the Biodiversity Unit. We have vast natural history collections, scientific natural history collections, and actually we have a very good collection of Baltic Sea samples also, and from the islands, for example, of the Archipelago Sea insect and spider faunas. We have tens of thousands of samples collected during different periods of time. Then we have the Botanical Garden, which is one of the most famous tourist attractions in the Turku area. We have also Aerobiology Unit, which is producing all the national pollen forecasts to the national TV and radio stations on a daily basis. And then we have Science and Luma centers, which are dedicated for children's science and environmental education. And then we have two important research stations. One of them is located in the northernmost Laponia, very close to the village of Utsjoki, and another is of course the Archipelago Research Institute in the island of Seili. And I suppose that most of you have already visited Seili, and I truly hope that if you haven't been able to visit Seili yet, you would have a possibility to visit the island of Seili in the future. The island of Seili is, in my opinion, is one of the most beautiful and one of the most diverse islands in the Archipelago Sea region. So it's a privilege to have a research station there. And still about 5 years ago, the future of Seili, or the Archipelago Research Institute, was uncertain. So, for example, the University of Turku was really thinking whether we should stay in the island of Seili and develop the Archipelago Research Institute also in the future, or whether we should leave the island. And luckily, the University of Turku made the decision that we will stay in the island of Seili. And in my opinion, it was one of the best decisions that University of Turku has made during the last few years. Of course, unfortunately, we have had to make some dramatic changes in the island of Seili and also in the research station. But most importantly, it is now clear that we will the University of Turku will be present in the island of Seili also in the future, and the marine research and marine biodiversity research is also highly elevated to the strategy of the University of Turku. So in the University of Turku, we have 6 profiling areas or thematic collaborations of research and education. And one of them is, of course, sea and maritime studies. It's one of those profiling areas that has been around already for some years. But now we have also a new profiling area, which is called biodiversity and sustainability. And it is also very important what comes to the marine biodiversity research. So in fact, out of 6 profiling areas, 2 are linked to biodiversity research at the moment. The sea and maritime studies, which we share with the Åbo Akademi University, and then the biodiversity and sustainability. And I have to say that I'm extremely proud of the University of Turku that we have this new profiling area called biodiversity and sustainability, because all the Finnish universities are actually profiling themselves into sustainability or sustainable development. But I suppose that the University of Turku is still the only one that is profiling itself also to biodiversity issues. So that is quite important and shows that we are really trying to develop new ways to study biodiversity also in the Archipelago Sea region. And why are we so interested on biodiversity? Of course, because biodiversity loss is one of those global challenges that is advancing at an accelerating pace at the moment. So, as you know, for the last 15 years or 20 years, climate change has been a very hot topic in both academia and in the society. But only during the last couple of years, biodiversity loss has become to be as important as climate change. And it's a very hot topic at the moment also in the society. And I'm extremely thankful to FINMARI also that you have selected biodiversity to be one of the topics in today's Researcher Day. I think that's a very important thing. So biodiversity loss is advancing at an accelerating pace, and at the moment we are already in a situation that the biodiversity loss has continued for such a long time that we are already using the term biodiversity crisis. So in a similar way than with the climate change, we are not only talking about climate change, but we are already using the term climate crisis. And in a similar way in the biodiversity issues, we are already, unfortunately, using the term biodiversity crisis. So the situation is quite difficult at the moment. But what is important to know, we can still stop the biodiversity crisis or biodiversity loss. So it is still possible to find ways to tackle biodiversity loss. if we don't do it now, then we will enter the era that will be called sixth mass extinction, and that will be really difficult, because at the moment we are already in risk of losing like 1 million species from Earth, and that is a lot if you think that there are altogether about 8 million species on Earth. We are in risk of losing 1 million species during the next couple of decades. And for example, the World Economic Forum showed the importance of biodiversity loss last year. They actually elevated the biodiversity loss among the most important global shapers, threatening the future of humankind also in the future. And at the moment, biodiversity loss is at the same level with, for example, with the weapons of mass destruction. So it tells you something about the critical situation with biodiversity. And we all know what are the direct drivers of biodiversity loss globally, but also, for example, in the Archipelago Sea region. We can actually see all of these global drivers functioning in the Archipelago Sea region also. So there are massive changes in land and sea use. There is direct exploitation of species. Climate change is threatening many species. There is loads of pollution, and also invasive alien species are threatening the species, the natural species of the Archipelago Sea region. And unfortunately, despite all the efforts and despite all the legal instruments, for example, the biodiversity loss is accelerating, and we are in risk of losing massive amounts of species, also in Finland, but also in the European level and also at global scale. And what comes to the biodiversity loss, I hope that we all understand that it's not only an environmental problem. It's really threatening the future of humankind, and for that reason, we should try to do everything in order to tackle biodiversity loss in the future. And of course, there is already good things happening. For example, the European Union published its new biodiversity strategy for the year 2030 a couple of years ago, and then they are now drafting the new regulation on nature restoration, which will be extremely important, for example, in the Archipelago Sea region also. And this year is very important what comes to biodiversity. We can talk about the biodiversity super year, because this year there will hopefully there will be the Kunming meeting, also the UN biodiversity conference, which will be held in China. And if everything goes well, it will be held in July or August. And in that meeting, they will draft also the global biodiversity goals, which will be probably quite similar with the EU level biodiversity goals. And what comes to the Archipelago Sea, I would say that Archipelago Sea is extremely interesting what comes to the biodiversity research. But I would like to challenge all the marine biodiversity researchers also, and all the researchers working in the Finnish coastal areas, to work even more in the Archipelago Sea region. Because at the moment we have, for example, 3 research stations in the Archipelago Sea region, but they are actually quite underused. So in my opinion, we have quite little research activities going on in the Archipelago Sea region at the moment. And this is a fact that we have been trying to promote, or also, for example, in collaboration with the Associate Professor Jari Hänninen last year. So we tried to elevate this fact that there should be more interest towards Archipelago Sea also in the academic world. So, for example, in the University of Turku or in the University of Åbo Academy, we have only a few research teams which are actually working in the Archipelago Sea region. And in my opinion, we are wasting very good possibilities, because at the same time, we have to remember that the Archipelago Sea region is one of the globally one of the most unique archipelagos in the world. And if you think that major part of the major breakthroughs, for example, in biodiversity research have been made in interesting archipelagos. So we should be more active in the Archipelago Sea region. We still need more basic information from the Archipelago Sea region. We need more species inventories and more monitoring information. And that is something that we are trying to develop at the moment also in the University of Turku. We are, for example, now inventorying the insect and spider faunas on many sandy esker islands in the Archipelago Sea region. And we have been working in those islands for a couple of years now as part of the Rannikko Life project. And we have already found incredible amount of new species for the Finnish fauna, which shows that we still know very little about the faunas of those islands. And at the same time, we have been able to verify the existence of a considerable number of very highly threatened species in those islands. And we are continuing that work now, and we are also trying to compare those new data for the materials or for the natural history collections that we are housing in the biodiversity unit research collections. And then we also need multidisciplinary research. And one example is the Living Island project that we are also coordinating in the biodiversity unit. In this project, we are trying to study the human nature or human biodiversity relationship in the Archipelago Sea region through the centuries. So we are combining archaeological history research and biodiversity research methodologies in order to understand a little bit better how the human biodiversity relationship has changed during the last few centuries in the Archipelago Sea region. And we are trying to find, by using that information, we are trying to find clues on how should we deal with biodiversity in the future. How should we affect the human biodiversity relationship in order to create a little bit better world for biodiversity. And then as another example of a very multidisciplinary project that is now happening in the Archipelago Sea region is the Beautiful Consortium. This is a STN funded 4.2 million euros funded project that we just started this year. And in this beautiful project, we are joining the forces of 4 different Finnish universities and research institutes in order to study the biodiversity respectful leadership. How should we lead people in the future in order to gain a little bit better world in the future? And most of the field works that are happening in this project are taking place in the Archipelago Sea in the future. So we will be active, for example, in the island of Seili, but also, for example, in the Kvitia farm in Parainen, and also in many other places, in many other islands in the Archipelago Sea region. But besides of research, I hope and I challenge all the researchers to promote their research in multiple ways. We should still remember that most of Finnish people, for example, do not understand well what is biodiversity loss and what biodiversity means actually. So besides of doing science, we should be active also in promoting the results of our scientific research. And that is something that we are also trying to do at the moment, and with many of our collaborators. For example, Forum Marinum showed a good example last year. They opened a very nice new exhibition on Archipelago Sea in Turku, Forum Marinum, and biodiversity is also very well presented in that exhibition. And it reaches tens of thousands of people annually. So it's a very good possibility to tell people why it is important to conserve the Archipelago Sea, why it is important to do scientific research, scientific biodiversity research in the Archipelago Sea region. And we are also opening a new science trail in the island of Seili next summer. So that is something that we are hoping that will draw the attention of the visitors to the biodiversity issues. So we receive about 25,000 visitors in the island of Saili annually, and with this science trail, which is also part of this Rannikko Life project, we are trying to show those visitors what kind of biodiversity research is being done in different parts of the Archipelago Sea region and why it is important. So I truly challenge you all to remember also the science communication side of your research. It is important that we promote our research for the big public also. So in summary, I have 1 minute left. In summary, I would like to challenge you to still enhance scientific biodiversity research into Archipelago Sea region. As said, in my opinion, Archipelago Sea is highly understudied at the moment. And we should, of course, when developing this research, we should remember that there are both aquatic habitats and also terrestrial habitats, and also there are a huge amount of people visiting and also living in the Archipelago Sea. So it's a good possibility for multidisciplinary biodiversity research also. We should also remember to mainstream our research in order to make biodiversity and biodiversity loss a little bit better known among the big public. And in my opinion, we could actually also try to promote Archipelago Sea as an international pilot area in tackling biodiversity loss, not in tackling biodiversity as you said in my slide, but in tackling biodiversity loss. So with these words, I would like to thank you for your attention, and I would like to wish you all a very good and safe field season in the Baltic Sea, which is starting in these days. Thank you.
Thank you very much, Ilari. That was a good presentation and very interesting things to come. Personally, I love it when you said about this promotion and science communication. As a communication person, I find it very important. It's something that should be the backbone of everything, that once we have good work, we have results, then we spread them. Otherwise, it's a wasted opportunity. And those actions sound very innovative. The science trail and all those Forum Marinum things, they sound very good, and all the best for those. At this point, we don't have any questions in the chat, but please use the chat for the questions that we will then raise in the Q&A session after this final afternoon session. But yes, please.
There is actually a question in the chat for Ilari. It's Erik Bunstolf is saying that I disagree that the work is not done in the Archipelago Sea. For example, the vast Velmo project and the fantastic work on thousands of islands and birds and plants. So maybe wondering maybe if Ilari is aware of all that underwater work that is prepared.
Yeah, maybe I used a little bit strong words in saying that. Of course, there is biodiversity risk being done in the Archipelago Sea region, but I truly challenge that we should have more research going on in the Archipelago Sea region. As said, for example, in the University of Turku, we have only a couple of research teams working in the Archipelago Sea region. And as said, it is very important to notice that I'm not only talking about a basic biodiversity research, but I'm also talking about all the multidisciplinary possibilities that this unique archipelago offers us. So in my opinion, I think that we still have a long way to go, and we have truly remarkable possibilities to develop many kind of biodiversity research, not only biological biodiversity research.
Thank you, Ilari. And as we speak, there is also another question in the in the chat, and we still have time for that. Meri, our morning host, is agreeing with Ilari. This really is a biodiversity super year. But then, on the other hand, the horrific situation in Ukraine draws our attention to basic things like food, energy, security of supply, etc. Do you, Ilari, think that there is still room for biodiversity issues on the public discussion, or is there any possibility to combine these topics?
Yeah, the situation is very difficult at the moment, and I have been also thinking a lot, because I'm quite active in science communication also, and I have been actually halting all my activities during the last few weeks, so because I have thought that it is very important to give all the attention to Ukrainian to the war situation. So the situation is difficult, but I truly hope that at the same time, little by little, we will be able to bring more information again on biodiversity loss and on climate change also, because those are the two global crises that are happening at the same time, and they are actually making the situation even worse all the time. And I think that there are some activities already that are joining, for example, studying climate change and crisis situations at the same time. But in biodiversity loss, there is a huge possibility also to think those situations at the same time. So I truly hope that we will see that kind of research coming out very soon. But at the moment, the situation is very difficult, and as said, for example, myself, I have been very careful in posting any, for example, Twitter messages about biodiversity during these weeks. I think that we should give as much attention as possible to the Ukrainian difficult situation, and then little by little we should elevate again, for example, biodiversity issues.
Thank you, Ilari. That's a good comment on that. We'll move forward. There are more questions in the chat, but we have time for those when we reach the Q&A session. So now let's move to the next presentation. Riikka Vainio from University of Turku is talking about trophic dynamics of mercury in the Archipelago seafood web. So welcome, Riikka.
Hello, my name is Riikka Vainio. I'm from the University of Turku, where I'm doing currently my PhD. And today the topic of my presentation is trophic dynamics of mercury in the Archipelago Sea. So mercury is a global environmental problem. It is released into the environment from natural sources, but in larger amount from human activities, such as different industrial processes. And even though emissions of mercury have decreased locally, for example, in Europe, the global emissions are still actually on the increase. And for example, the global atmospheric emissions have been estimated to be increased 20% from year 2010 to 2015. And in the environment, mercury is persistent, bioaccumulative, and toxic to organisms. And the cyclation and chemistry of mercury in the environment is quite complex and affected by both abiotic and biotic factors. Mercury can be transported over long distances via atmospheric currents and deposited far away from the actual source of contamination. And in aquatic systems, the differences in chemical, physical, and also biological processes between the water column and the sedimentation or the sediments can lead to differences in concentrations and bioavailability of mercury in these environments. And this in turn leads to that species habiting different environments and feeding from different sources can be exposed to different amounts of mercury during their lifetime and through different trophic pathways. And as I already said, mercury is bioaccumulative and toxic to organisms, and it's also biomagnifying in the food chains, meaning that the mercury concentrations increase along with the trophic level, with highest concentrations found in apex species. Mercury is also present in the Baltic environment in relevant concentrations. This map shows in the reddish color the areas where mercury concentrations in biota, in this case fish, exceed the limit determined for good environmental status, and this limit has been determined in the European Union Water Directive. So mercury is present in the Baltic environment, but despite this, the trophic dynamics of mercury and the trophic pathways of it are not very well known. There have been few studies investigating the trophic dynamics of mercury, but these have been focused on very simple systems and simple food chains with only few species within them. And this, of course, doesn't reflect the reality very well, as this is a complex food web consisting of species with different physiologies and different ecological factors. And this leads to that there are different pathways through the trophic system which mercury can be transferred. So we set out to investigate the trophic dynamics of mercury in the Archipelago Sea, located in southwestern Finland. And in this map, you can see the sampling areas. And here I will show you the system we sampled. So we divided this system or this species to benthic and pelagic. And first, there is the pelagic part of the food web. On the green, you can see the primary producers, and on yellow, the invertebrates. And the invertebrates vary in diet from omnivory to herbivory. Then on blue boxes, you can see the benthic fish, with diets also ranging from invertebrate to mixed fish and invertebrate diet. And then, in addition to the benthic ones, there are the pelagic species, and the base of the pelagic food web or the food chain consists of zooplankton and phytoplankton. And then there are the fish feeding on those, and again, there are predatory fish feeding on the other lower trophic level fish. But in addition to the solely benthic and pelagic species, there are also species that can move between these two environments and feed from both pelagic and benthic sources. And we categorize these as bentopelagic species. And these are part of both the benthic and pelagic food chains. And we also sampled 3 bird species. First, there is the common eider, which is a benthic feeder with bivalve diet. And then there are white-tailed eagle and great cormorant, which are bentopelagic species, great cormorant feeding on fish and white-tailed eagle feeding on fish and the other birds. And out of these samples, we analyzed the total mercury and stable isotopes of nitrogen. And we used the stable nitrogen isotope signal to estimate the trophic position for each of these species. And then we modeled the relationship of logarithmic mercury concentration and the relative trophic position for the benthic and pelagic food chains with a linear model. And from the slope of the linear model, we calculated the trophic magnification factor, the TMF, which estimates the average fold increase in mercury concentration across one trophic level. And we left the primary producers out of these models because their stable nitrogen isotope signal can show considerable temporal and spatial variation, which we couldn't account for. And I will quickly show you the results for the trophic positions. So on the left, the benthic food chain, and on the right, the pelagic food chain. And on the orange, you can see the invertebrate species towards the bottom of the food chains. And then towards the top are with blue the fish and with red the birds. And you can see that the birds are actually located not directly on the top, but rather towards the middle in the mixed between those higher trophic level fish, which are pike, forehand scalping, and the ruff. And then the results for the trophic magnification models. So first I will show you the model including all the species. So the shape of the point tells you which kind of species it is, and the color tells the food chain. So orange are the benthic species, blue are the pelagic species, and red are the bentopelagic species. And the bentopelagic species are included in both benthic and pelagic food chains. And the dashed line is for the benthic food chain, and the solid line is for the pelagic food chain. And you can see that the slope is steeper for the pelagic food chain, and the TMF is also higher. But actually, there is no statistical difference between these two slopes in this model. And here you can see circled three points, which are the bird species. And you can see that their mercury concentrations are higher in relation to their trophic position than for the other species. And they don't seem to fit the models quite as well as the fish and the invertebrates. And similar results have actually been found also on other systems and on other contaminants. And it has been accounted for due to physiological and ecological differences. And birds have, for example, higher energy requirements than the poikilothermic fish and invertebrates, and thus higher consumption of prey and contaminants. So, because the birds don't seem to fit the model quite as well as the other species, we modeled the same system also without them. And here you can see that this looks quite similar, with the pelagic line being steeper and the trophic magnification factor being higher. And without the birds, this is actually a significant difference between these two lines and food chains. So the trophic magnification of mercury seems to be higher in the pelagic food chain of the Archipelago Sea compared to the benthic one. And as I already told you, these are very complex systems, and it's hard to exactly pinpoint why this difference exists. But it's probably tied to different speciation and bioavailability of mercury between these two systems. And it would be very interesting to, in the future, further investigate why this difference actually exists and maybe look into the speciation a little bit further. So again, as a summary, higher trophic magnification in the pelagic food chain, meaning that concentrations increase faster per trophic level or more per trophic level in the pelagic food chain. And part of these samples were collected with help of Saley Archipelago Research Institute and Tvarminnet Zoological Station. And I would also like to thank other people who helped with the sample collection and fieldwork, as well as my funders. Thank you.
Well, thank you, Riikka, for this interesting talk. Do remember, everyone, to add your questions to the chat. And as our next speaker, we will still be looking at the food webs of the Archipelago Sea. Now it's Arne Lauerma from the University of Turku and Saley, who will be talking about the blue mussel color polymorphisms and growth rates in the Archipelago Sea. So please, Arne, the floor is yours.
Hello, my name is Arne Lauerma, and today I'm going to be talking to you about the blue mussel color polymorphism and growth rates in the Archipelago Sea. So, first, a brief introduction to the blue mussel. As we know, it's a key species of the Baltic Sea, mainly because its role in biomass and nutrient cycles, as it's the most prominent filter feeder of the Baltic Sea. It's also an important food source for many species like eider and flounders. And as a habitat-forming species, it's important to other species of the seafloor as well. Our mussels here at the Baltic Sea are a genetic cross between the Pacific and the Atlantic blue mussel, which makes them slightly globally unique. And as a marine species living in the Archipelago Sea brackish water conditions, individuals here are under severe salinity stress, which comes at an energetic cost which means that our individuals here remain small compared to their marine conspecifics at least. The blue mussel is polymorphic in terms of its shell color. The shell coloration is determined by a single locus with two alleles, with the brown allele being dominant and the more traditional dark blue being the recessive of the two alleles. All the dark blue individuals are relatively the same color, dark blue, but inside the brown morph there is considerable variation. And individuals in this picture we can see a fairly light one, but they can be significantly darker as well. They can also exhibit stripes, which leads to thinking that maybe there's a different loci which also affects the coloration. In growth rates which were done in the Atlantic coast on Mytilus edulis, they found a significant growth rate difference between the color morphs. The brown morph was growing at a significantly slower rate, which was theorized to be related to the thermoregulation of the animal, as blue mussel is a cold-blooded animal, and if darker color means that they can absorb the sunlight better, they may have a higher metabolism in the cool conditions. So we set out to sample the population of the Archipelago Sea, and we wanted to get mussels from variable sites with variable conditions. We ended up with these 4 spots, 2 of which are in the outer archipelago, 2 in the middle to intern archipelago. We did the sample collection with an epibenthic dredge and, in one instance, diving. We cleaned the mussels and brought them to lab conditions where the age of the mussels was determined, which is done by counting the winter grooves on the shell. On the picture we can see some of the grooves, and we can see that the animal is grooved, the shell is grooved all over, but the winter grooves, which are formed in the winter when the growth is stopped, are much more prominent. After that, the growth rate was calculated by dividing the length of the mussel with its age. And in total, we analyzed 524 individuals in this manner. For statistical analysis, we did 1- and 2-way variance analysis. Next to our results. And here are the results. First thing to note is that we found a relatively even distribution of color morphs at every site. There's a significant amount of brown morph everywhere, but the blue morph is slightly more prevalent at each site. Next thing to look at here is the growth rates on the right. And we found that the growth rates were really similar between the morphs at every site, almost identical. Something to note here is also that the standard deviation of the data is notable, as there are large individual growth differences between mussels. Here on the graph we can see it a bit better. The differences between color morphs are quite small, at least compared to the standard deviation. We found one significant effect on growth, which was location, which is not really that surprising as our spots varied in environmental factors like salinity and sample depth. Also, the nutrient concentration of seawater may be different across these sites. That was not measured. The slowest growth rate was found at the innermost spot, Grundet, which has a relatively low salinity for blue mussel, and also it is notably the shallowest sample spot we had, which may be disadvantageous in summer months if there is heat stress. The outermost spots, Voodoo and Buttonshare, had the best growth rates. Again, not that surprising. But again, the color morphs were relatively even everywhere. So I have to say that we did not find a significant difference between color morphs here. It seems like a growth rate difference doesn't exist, at least in the same magnitude as it does in the Atlantic Sea conditions. If there is a smaller difference between these morphs, the high individual variation makes observing it quite difficult. We found no real aerial trends, as the percentages of brown and blue morphs were relatively the same at every spot. Going back to the thermoregulation hypothesis, it suggests that dark blue individuals should do better in the darker and colder conditions, and Archipelago Sea can be read as part of these conditions. Also, the turbidity increase of the Archipelago Sea in the past decades should be a factor which would favor the darker morph if thermoregulation is the deciding factor between these two morphs. However, the sentiment of the local people is that the brown morph is the one which has gotten more common in recent memory. And this presents the underlying question, why is this? And I have to say that we don't know. We don't have a good answer yet. But what we do know is that the mussel population of the Archipelago Sea is threatened by environmental changes, as I felt personally when we tried to find mussel in the inner archipelago. Increased sedimentation is destroying their preferred habitats. Decreasing salinity increases salinity stress, and for the shallower spots, increases in the water temperature may increase summer heat stress and mortality related to that. At present time, we do not know if the morphs have a real difference in tolerance to these changes, but with them continuing at the Archipelago Sea, we may know in the future. Now I would like to say thank you for listening to me, and a special thank you to the Nauru Rotary Club for your support. Thank you.
Thank you very much, Arne. And we'll continue straight to the final speaker before the final discussion and final Q&A. So we are reaching the end of this wonderful day. But before that, I'd like to present the next speaker, who is Laura Harris from the University of Maryland. And Laura is in Finland as a Fulbright scholar this year and is spending several months at Tvärminne. But right now, however, Laura is back in the US and joins us from there to talk about her recent research, which is called Making Space Matter for Hybrid Ecosystem Individual-Based Models. So, Laura, welcome.
Hello, and thanks so much for having me in this seminar today. My name's Laura Harris, and I'm currently back in Maryland at the University of Maryland Center for Environmental Science, but I recently left Finland, where I was hosted at the Tvärminne Zoological Station by Dr. Alexandra Lewandowska as part of my Fulbright Finland Seeking Solutions to Global Challenges Award. And while I was in Finland, I was able to really jumpstart a program where we were thinking about comparisons between the Baltic and the Chesapeake Bay, but I won't have time to talk about that, and it's really just starting, so stay tuned. But I did want to mention this really valuable work and all the wonderful people who I'm engaging with around this that have been so nice to get to know better during my mini sabbatical this winter and spring. But today I'm really going to be focused on sharing another part of my research, which is using numerical models to simulate estuarine and coastal ecosystems. And in particular, I want to spend some time today thinking about the use of spatially explicit individual-based models or agent-based models. A lot of these have been applied to aquatic ecosystems, and there's a few of them here in this slide, in particular to simulate fish or larval movement. And it really makes sense to use this numerical modeling approach for organisms that move, because they focus on creating a set of very simple rules that hopefully lead to some emergent behavior in the model dynamics that will help us have a better understanding of ecology. In the coastal environment, it's much more common for us to use this type of a modeling approach. This is a conceptual diagram used to help constrain a numerical model that my colleague Mark Brush created for a shallow estuarine ecosystem. And these are kind of an upgrade of oceanographic MPZD models. We're really simulating state variables with units of grams carbon per meter squared or per cubic meter, and thinking about all of the different environmental factors that influence the fluxes or the rates that connect all of these different state variables. And this is a wonderful approach. I love these models, but it really doesn't borrow as much of the learning as we've had from the more terrestrial and ecological world. And so one question is whether we might blend some of these approaches together and think about whether IBMs might be valid in aquatic ecosystems. And one of the reasons I started to think about this over a decade ago was looking at organisms like eelgrass. This is a seagrass, Zostra marina, that you also have here in the Baltic. And when I was thinking about writing a model for this and looking at existing models, many of them use this state variable grams carbon per meter squared approach. But when you look at Zostra, it's so clear that it's a plant. It has different modules in terms of the ramets and the rhizomes, and that it also essentially moves by colonizing the benthos. And this image on the right is actually a drawing that was made in the San Francisco Bay Estuary in the 1920s, tracking mother and daughter ramets through a large, spatially extensive eelgrass meadow. So this is something I think we've lost a little bit of thinking about the botany of these plants. The other thing that I think is helpful to think about in terms of IBMs for coastal ecosystems is that there's a lot of feedbacks on ecosystems that are spatially explicit, and organisms like oysters in particular can have a pretty dramatic impact on their local environment. So for example, the photos at the top on the right are a tank of oysters that we add some phytoplankton-rich water to, and the image on the left is that same tank just an hour later. And so these oysters have this phenomenal impact on the water column. But they also interact in a very individual way around the hydrodynamics of the system. So the images on the bottom are field data we took from longitudinal field data from over a restored oyster reef, where we could detect this pattern of gradients in particle concentrations depending on the tidal direction. That gave us some ideas of how the rough, chunky oysters on the benthos were interacting with the tide and the hydrodynamic forces. Now, layered on top of this is that IBMs have a rich history of heuristics and how we should be using them. There's kind of sets of rules. We should include life history. We want to think about resource dynamics. Fundamentally, we want to be able to model individuals discretely. And all this creates kind of a bottom-up approach for testing our ideas around emergent behavior and why that emergent behavior might happen in these ecosystems. And that's a critical kind of intellectual approach for doing this that I think is really valuable for these ecosystems as well. And so I'm going to breeze through these two examples where we've done this in an eelgrass meadow and then in an oyster reef. And just to highlight to you that, especially for eelgrass, this is an organism that has different life history stages. In the Baltic, it's largely clonal, so you kind of miss some of the sexual reproductive aspects of eelgrass, but nevertheless, that's a critical part of it in many other parts of the world. And we created this virtual eelgrass meadow to both include that and to have rules about when the seed bank germinates, the specific growth rates that the seedlings have that are different ontogenetically from the adults, thinking about how biomass is allocated between roots and rhizomes and leaves, and when they flower in warmer, lower latitude locations to create this IBM for a virtual eelgrass meadow. All of this relied on really thinking about plasticrone intervals for eelgrass. The plasticrone interval is the interval between leaves in this meristematically growing plant. And it's a nice way of kind of timing the way that the clone lays down. If you think of my bicep here as kind of the rhizome, and this is the leaf, the clonal plant is kind of laying down that material over the benthos. And all of that gets tied to the meristematic growth and how leaves are produced in the plant. So we simulate all those rules. These are essentially IBM-based rules for that. It kind of became our leaf clock for branching rates and ended up resulting in shifts in shoot density that we could then have feedbacks on available light. So there was a circular feedback integrated into the model. And we eventually were able to do lots of simulations at different densities, which allowed us to use a really well-known plant universal law of self-thinning to test whether the IBM was valid. And this is always a tricky thing in IBMs because they're so complicated, is how do we validate them? So here's some red data, our empirical data from mesocosms, and the green are from the model simulation. So we did a pretty good job. And quickly now to the second case study for what we've done with oysters. We've created a model called Reef Biodes, and this is really kind of different modules. The first module has the most IBM characteristics, where we simulate flow over an oyster reef where each of the oysters are individually modeled and can each have their own size. And it's linked to an Advective diffusion model grid, and all of that gets tied into biodeposition and nitrogen cycling. So I'll step through this very briefly. The Advective diffusion model simulates advection of these particles as a function of vertical turbulent diffusivity, and then removal, where that's really filtration that we're talking about. And that filtration is an allometric formulation that's related to the individual sizes of those oysters and also connected to individual bioenergetics for each oyster so that we can simulate their growth, and they're all changing over time as the model simulation progresses. And all of these are a function of temperature, salinity, and how much food is in the water. That's then connected to production of biodeposits and a really detailed hydrodynamic set of formulations that I won't go into here about how resuspension happens around the shape and the individual kind of morphology of each of these oysters. So kind of very fussy IBM details. And then all of those biodeposits essentially feed a biogeochemical model, the sediment flux model, to simulate nitrogen dynamics and to think about the denitrification or nitrogen removal capacity of the reef, as well as its ability to recycle nitrogen. And we were able to use this model to simulate the patterns we were seeing in our field data, so that was wonderful. And more importantly, we were also able to think about, well, using this model to really in fine detail consider what chlorophyll removal capacity you might have in different parts of an ecosystem. And we're hopeful this will be used for restoration and perhaps aquaculture plans in the future. So to finalize very quickly, I think we can really have a lot of fun Using these modeling approaches, it allows us to think creatively about movement, for example, clonal growth. It reminds us that aquatic organisms aren't a black box of carbon. We have to think outside of that black box to remember that size matters and that feedbacks of the spatial environment matter. And all of that is really fostered by this IBM mentality, in my opinion. So I'll just end, because I know I'm running out of time, by acknowledging Kevin Kahover and Jessica Foley, who were two master's students who worked on these models, really phenomenal master's students, and my funding sources. And I'm really happy to be here with you and take any questions. Thank you.
Well, thank you, Laura, for this presentation. And I would just like to sort of comment here directly that it's really nice to see that biodiversity is not just about what species we have, but also understanding what their role is in how the ecosystem functions. So that was very nice that you could join us from the States. Hopefully your jet lag is not too bad. And then I guess we're moving on to the last questions session. How are we doing? Do we get the do we have the other speakers back online?
I guess we have almost everybody except Ilari. So Ilari, if you can hear us. Wonderful. Yes,
but also understanding what their role is in how the ecosystem functions. So that was very nice that you could join us from the States. Hopefully your jet lag is not too bad. And then I guess we're moving on to the last questions session.
Sorry,
how are we doing? Do we get do we have the other speakers back online?
I guess we have almost everybody except Ilari.
I'm here.
Please turn off your stream.
Okay, hopefully now everybody is here with us. Every speaker of this session, and hopefully no one has the stream open because the sound comes a little bit later, so it's almost impossible to speak and listen to the stream at the same time. So the final part is the Q&A, and Joanna, should we start with some questions to Ilari? Ilari has been very active in chat, but maybe if there is someone who is unable to check the chat and is only listening, maybe we could wrap up this discussion a little.
Yeah, there's been an intense and interesting discussion in the chat, so I do suggest that everyone who has the chance to go and read that through, because we won't read all the questions out there, but we'll pick a few ones. For example, Roxana Preston, who is a PhD student at the University of Helsinki, herself working on genetics of bladderwrack, is asking Ilari that biodiversity is often considered just under the scale of species diversity, yet biodiversity ranges across several scales. Are you aware of any research projects in the Archipelago Sea focusing on the other scales of diversity, for example, genetics? So if Ilari wants to repeat some of his answers that he's already put in the chat so that everyone can take part of that.
Yeah, so I already answered something to the chat, but I totally agree with Roxana that we are still using, many times talking only about species diversity in case of biodiversity. And I just mentioned in the chat that I have been thinking that we should even try to revisit the whole concept of biodiversity, because I would like to see much more human biodiversity interaction also included to the concept that it is in the traditional way. But there certainly is also interesting research going on different levels of biodiversity, including genetic diversity. And I just mentioned that there are some really interesting studies going on food webs, for example, in the Archipelago Sea region. So much research is going on, of course.
Thank you. Then there was a little bit of discussion that I would personally interpret maybe as everyone knows mainly most about what they're working on themselves. And Alf Norco was commenting here that biodiversity is an integral part of all ecology, and most marine ecologists in the Baltic have been working on biodiversity for decades and communicated this. And that marine science is inherited interdisciplinary, but of course that we would need to do more about understanding, for example, the links between terrestrial and marine biodiversity. And the direct question here, which I think is a really good one, is that do you, Ilaria, have any ideas about how to integrate these two realms, the marine and the terrestrial realms, for more holistic biodiversity analysis that could support sustainable development?
Yeah, and I answered that there, for example, we will have a new report coming from the Finnish Nature Panel, which is, and that work will be led by Christopher Boström from the Åbo Academy University. And I know that it will include also some of the top marine biodiversity researchers of the Finnish marine community. So I truly hope that, for example, in that report, we will see some examples of that kind of research that could join these two realms. And then I also said that I'm very much looking into the direction of this multidisciplinary research paths, joining the forces of natural and human sciences in order to find very good ways to advance sustainable development in the Archipelago Sea region. And that is, of course, something that we are trying at the moment. For example, in that beautiful consortium, we have economists, we have social scientists, we have biodiversity researchers, and they are all working together in order to create that kind of examples that could be used in order to promote the systemic transformation that we need in order to tackle biodiversity loss.
Yeah, thank you. And then actually I have one general question also of you, Ilaria, that you've worked in the tropics where species richness is just extremely high and you keep finding new species, etc. Whereas in the Baltic Sea we have rather low diversity and we're quite often used to being sort of apologetic about it. And that then our biodiversity research is maybe not of that general interest in the rest of the world. Do you have any comments on how we could change this thinking around?
Yeah, I think that we should think both terrestrial and marine habitat at the same time. So actually the terrestrial biodiversity is at highest level in the Archipelago Sea region in Finland. So we have very high numbers of species, for example, of many insect groups in the Archipelago Sea region. So Archipelago Sea region, although the marine habitats may not be so diverse as their counterparts in many other parts of the world, but the terrestrial habitats are actually extremely rich in species. And we still know very little about that diversity. For example, in the Archipelago Sea region, only a few insect groups have been inventoried during the last couple of decades. Most of the research has been concentrated on Lepidoptera, on butterflies and moths, and on beetles, on Coleoptera order. But all the other megadiverse insect orders, for example, have not been studied in the Archipelago Sea region. So there is a huge possibility to find even new species for science in the Archipelago Sea region if we look at if we take into account also the terrestrial habitats that are very diverse in the area.
Okay, thank you. Laura, do you have any other questions to pick up from the chat there?
Yes, there are a couple of questions to Arne. So let's start with Maiju Lehtiniemi's question. Arne, you mentioned factors that are threatening the blue mussels under changing environmental conditions. What do you think of introduced species like zebra mussel and Conrad's false mussel that are spreading on the Finnish coastal habitats? Do you see them as a threat?
Yeah, that's a good point, and I do see them as a threat. I have read reports of the zebra mussel decreasing numbers of the blue mussel in some conditions. At least in the lower salinity areas, they are definitely a threat.
Okay, and another question from Roxana Preston. For you, Arne, since the blue mussel in the Baltic Sea is a species complex, could this play a role in why you are not observing the same trends as in the Atlantic M. edulis population?
Yeah, that could factor into it. It would be really interesting to do like a growing experiment with our blue mussels.
Okay, thank you.
And then
Joanna, do you have anything?
I have also one more question for Laura. You work in the Chesapeake Bay, so there's lots of similarities between the Baltic and then the Chesapeake, and then of course differences. So, for example, we don't have any oysters, but we have other things. So, how now that you sort of been looking at us from outside a little bit, do you see any sort of major sort of gaps in Baltic Sea marine science from your point of view, or any areas where we are doing rather well in comparison with your home puddle?
Well, I'd say on this topic of biodiversity that the Baltic feels very much ahead of us. And that's, I believe, largely because of the regulatory structures. A lot of our research funding follows management needs in the Chesapeake region. And we don't have the same regulatory framework around biodiversity that is so strong in the Baltic. So, you know, that's something I would love to reflect on more with all of you, is that trickle down of what happens when, you know, the policymakers have decided that that's a priority for the community, and then that eventually impacts what research is funded. Rather than gaps, I'd say there's exciting opportunities. You know, your earlier question around how to better align biodiversity research with other ecological questions, to me, the obvious answer to that is always rates. As an ecosystem scientist, that's really the currencies we think in are around rates. And I know in the Coast Clim project that you all are doing that's a big part of things. But, you know, throughout the talks here today, I've been thinking like, oh, it would be great if along with those biodiversity metrics, you also had photosynthesis or respiration or secondary productivity or something that could then more easily connect with the ecosystem scale processes. So not a gap, but an opportunity.
Thank you very much. Do we have any more questions there? Checking the chat.
Not in the chat, but now is a good time to also give an opportunity for all the presenters we have here together. That do you have anything you would like to ask from each another?
I suppose no.
Everything is clear.
Everything is clear. Yeah.
Yeah. Okay. Well then I think it's time to move on to the final thanks and final words, etc., etc. First of all, I'd like to remind you all about the recording that will be available from the FINMARI website, and also about the feedback form. So all of you that have been registered to this event, you will get an email afterwards, and there's a feedback form, and we would really appreciate if you could give us some feedback so we can develop the event even further. I at this stage already, I'd like to say big thanks to everybody who has been speaking today, giving good presentations, very informative, lots of new knowledge, especially for me and hopefully also for others as well. And what else, Joanna? Any thank you words?
Yeah, before I hand over to Maju Lehtiniemi for the final thank you, I would also like to, on my behalf, thank all the speakers for a very interesting day today, the whole day. I think it's really valuable to get a bit of an overview of what's happening in marine science in Finland, but also from different fields and different disciplines, but also getting some input and insights from other outside our fairly close-knit community. So that's been really, really good. And every time not only Laura, but I think we all learn new things while listening to other researchers and other research groups talk. So that's been really valuable. And I would also like to thank everyone who's been listening in today. Thank you for the activities in the chat with the questions and the comments. And yes, please again have a look at the posters. There's quite a few, and from normal research projects, but also advertising a few new initiatives, new projects starting, and also, as I mentioned in the chat, there's a chance of still this week signing up for a scientific diving course. So that's also more info in there. So please look at all those posters as well. But yeah, thank you very much for an interesting day. And then I would just like to hand over to Maju then for the closing words.
Thank you, Joanna. So now it's time for the final thanks, or how was it? So the closing up of the day. First, I want to thank the session chairs, Joanna and Laura. You did a wonderful job in leading the discussions, so thank you very much. We have had a very long but really interesting day with various topics in the talks as well as in the posters. I really hope that you all in the audience also went to see the posters. They were a good addition to the talks, and nice ideas and results there as well. So it was really nice to hear all the talks and see all the results, and really got the impression again that we have so differing projects going on. It's important that we gather together at least once a year. We could of course do this more often, but at least once a year to hear what is going on and what are the newest things that we are working on. And these presentations also showed the importance of marine infrastructure and the various possibilities that we have for collaboration, new collaboration as well. So as said in previous presentations as well, I think we really have time now to think of new collaborative ideas. At least I got some new ones when listening to the talks today. So I really hope that we will have more collaborative projects in the future, and of course continuing the ones that we have right now, because we have really good infrastructure around the coast of Finland, and some unique aspects in different infrastructures. So it's possible to really join other infras and start some new collaborative projects in the future. So if you got any new ideas, just email the researchers and take contact. Then I would like to thank especially the keynote speakers of today, Sanna, Henry, Saara, and Ilari, for your valuable input to the seminar. It was really enjoyable to have some fresh angles from outside the FINMARI community. It's very important for us not to just, although this is a Marine Researcher Day, it's good to hear some words from outside as well. And like in the last QA session, it was discussed that, for example, this multidisciplinary research project would be needed, which would tackle both habitats underwater and in the terrestrial side. So yeah, very nice talks, thank you. And some special words for all the keynotes, starting from Sanna. I think it was necessary for us to hear how you see FINMARI to fit to the environmental infrastructure landscape in Finland and also in Europe, and also to get ideas how we could improve our positioning and networks in the landscape in the future. And of course, I'm also grateful on behalf of the whole FINMARI consortium to Sanna's last words when you were replying to Mary's question, saying that FINMARI is unique and kind of diamond marine research infrastructure, as it's not trivial. Not in all European countries, marine research institutes haven't been united like we have in Finland. So thank you for those words. But of course, there is always room for improvements. And as it was pointed out, we should work on the accessibility of our infrastructure platforms and data as well, of course. And this work is in progress. We have started, for example, to publish gear gallery sheets on the FINMARI websites, which are for everybody to see what equipment, what infrastructure we have in different institutes. And there are photos and some bullet point introduction to the equipment and also contact information. So that is a starting point, and in the end it's the idea that all our marine infra is presented there for people to see and also to take contact if you need certain equipment or would like to come to do research in our institutes. But in addition to this, we have to of course work more for the live and remote access of the various platforms that we have, that we are maintaining and also developing in FINMARI. Then to move on to Henry's keynote talk, it was also very inspiring to hear about the world of Argo floats, to see how large the network already is in the Baltic, and also about the future possibilities, what is coming up. So thank you very much for that. I'm sure many of us in the audience got new ideas and are going to take contact with some collaboration ideas and possibilities with you in the Argo float world. Thank you, Saara, for the talk on the Carbon Action Platform. And thanks very much for joining us this week on a very short notice. It was important that you could join, and your talk fit perfectly to the day's agenda. And it gave us a view from the terrestrial side, from the agricultural land, where decisions made, of course, affect the Baltic Sea directly. And at least for me, the talk gave kind of a positive feeling of the future, showing that if we make correct decisions and actions in the agricultural fields, we have a better future. We have a possibility to affect the nutrient leaching from the fields. And then thank you, Ilari, for your very timely talk on the biodiversity research and biodiversity crisis we are facing. This talk also gave us proof for the FINMARI consortium that we chose wisely when we selected biodiversity loss as the other science theme, in addition to climate change in the present work we do. So we got some new ideas from your keynote talk. So thanks for that as well. So, and as said before, and also as Ilari pointed out, we should increase the collaboration. So kind of now in the end, I would really wish that everybody thinks of new ideas, new collaborative things that we could do together and join forces, not only on the biodiversity side, but of course in all aspects of marine research. And then last but not least, I also want to thank the whole audience, everybody there who are listening, still listening, and not just in the morning, and have been asking questions. It has been very nice to follow the discussions. Tomorrow we will meet the Scientific Advisory Board and National Board of FINMARI for feedback. So we will hear feedback of our progress during the past year and also about the event today, and hear how to improve our work more. So that's important as well. So for the rest of the audience, thanks again. Thanks for all the talks, questions, and have a nice evening.
Thank you, Maiju. I believe it's 1 minute left. Any last minute jokes, Jan?
I think jokes are gone for today, so it's the final. Thank you, goodbye. That's that we have left.
That's the thing. Thank you very much, and bye-bye.